Professor Ashok Jadhav, Head of the Chemistry Department. On behalf of our JSM College Alibag, District Raigad, Maharashtra, I welcome to one and all on today's topic, Analytical Method Validation, online national webinar. Our inspiration is the Danta Shikshan Mandal's President, Advocate Gautam Patil sir. Under his leadership, we are giving our best to the institution and our students. Our principal, Dr. Anil Patil sir, one of the best administrator among the Mumbai University colleges. Both are helping us to give the support for various activities in the college. Today's webinar is such an example. Friends and faculty members may be enlightened with this most important topic in recent trends. Large number of material is available for the topic related information. Analytical method for the quantitative analysis and used to solve the many problems that we face, so that any analyst or the chemist need to test their knowledge, the validity of the analytical method to solve the given problems. In short, what is the analytical method validation? So it's the process of justifying that analytical methods applied to solve the process, to solve the problem. Friends, today's speaker, Mr. Animesh Shivaji Salunke, is the most eminent personality in pharma hub industry of Hyderabad, Telangana. I request Dr. Jayashree Patil to give the introduction of today's resource person of this online national webinar on the topic Analytical Method Validation.
Thank you, sir. Am I audible?
Yes, audible, madam.
Okay. Good morning, all of you.
Good morning,
Today
madam.
it's my great honor to introduce today's speaker, Mr. Animesh Salunke, sir. Sir is a dynamic professional with 25 years of rich experience in analytical development, quality control, and technology transfer in reputed pharmaceuticals like Optimus Pharma, Incube Ethicals, Microlab, Cipla Limited, Strides Arcolab Limited, is site head in Vitron Advanced Analytics and Private Limited, analytical CRO based organization, Hyderabad since from June 2019. Sir is leading team of plus 75 from analytical developments, IVRT, Q3 characterization, peptide mapping and characterization, Q3 3D study, medical device testing. Sir has extensive method validation and new product testing. Sir has in-depth knowledge about compliance of various quality measures by maintenance of appropriate requisite documentation and records. He is proficient in stability of new formulation of new developed product as well as existing formulation as per ICH guidelines. Sir has an effective communicator with excellent relationship management skills and strong analytical problem solving and organizational abilities. Sir is certified as champion for implementation of Lean Six Sigma BMGI, that is Breakthrough Management Group of India, and actively participate in the implementation of Lean and 5S techniques in the plant. Sir has extensive experience in state-of-art analytical laboratory. There are so many notable achievements on his credit, but I am going to highlight some of them. First, to file ANDA project of a mefenic acid capsules, patent for trimetazidine dihydrochloride modified release tablets, new R&D lab setup for analytical departments including utilities and instrumentation. Sir has also set up of export-oriented QC laboratory for Ponda Goa unit, and Sir has FDA approval. Sir has got FDA approval in chemical and instrumentation. Today, such a nice personality we have as a resource person. Now I would like to request Salunke sir, please start the session.
Thank you, madam, and good morning, friends. First of all, thank you to principals, honorable principal sir, president, and all teaching staffs for providing me this opportunity to interact with all of you. I think analytical method validation is a vast subject, and I'm not sure how much I'm able to explain this virtual, because if it is offline, it is good, but I will try my best to understand you. So I'll share my screen. Just check it whether it is visible for you. Can you see the screen? Hello?
No, no Animesh, sir. Satish, sir, please.
Now it's okay?
Yes, sir.
Yes,
Yeah,
okay,
it's
okay. Now it's
visible.
Okay, once again, good morning. So this analytical method validations, am I audible to everybody?
Yes, sir.
Yes, sir.
So coming to this analytical method validation, maybe all of you have the multiple questions like what is method validation, why it is required, when it is required, and what are the products, what are the tests it is required. I will try to explain all these details in short. In between, if you have any questions, please stop me and you can ask, or we can have the question and answer section at the end also. So this analytical method validation, there are multiple types of tests are there, like related substances for impurity profiling, then potency, that is purity of the particular product, whether it is solid oral or whether it is liquid oral or ophthalmic products, any product. Method validation is applicable for all the products which are marketed in Indian market, whether it is US market, whether it is European market or any other country. It is applicable for all the markets. And there are many guidelines are available. I will give the references at the end of this presentation. You can note down the references. Guidelines, there are little restrictions we are having. So guideline is not vast guidelines, but based on the experience. Sorry, in between some voice.
Kaushal, unmute yourself. Kaushal, yes, okay.
So there are many guidelines are available in the industry, or you can refer in the internet also. Each guideline have little different requirements, but based on the experience in last 25 years, the different country has different expectations. Like US country has different expectation, European has different, ICH has different. Finally, they have done the harmonization, and it has come in the ICH guideline. It is called as International Conference on Harmonization. So they have harmonized all the guidelines now. So each and every country requirement is almost similar. So this validation is applicable for qualitative testing as well as quantitative testing. So, but the parameters will vary, depends upon the method, depends upon the test parameter, and depends upon the techniques. There are different techniques are there, like titration or UV spectrophotometer, or you can have this gas chromatography, high performance liquid chromatography, liquid chromatography with mass spectrophotometer. So there are many instruments, many techniques are there. Based on the instrument and based on the techniques, the parameters for method validation will vary. So considering this, I think we can start. So first of all, what is the analytical method validation? Maybe everybody is having this question. So what is the validation? So you can see this is the basic definition for validation we are having. So validation of an analytical procedure is the process by which it is established by laboratory studies that the performance characteristics of the procedure meet the requirements of the intended analytical application. So there are different definitions you will find in different guidelines. You can see the another definition. Method validation means establishing through documented evidence a high degree of assurance that an analytical method will consistently yield results that accurately reflect the quality characteristics of the product tested. So if you go to the different references on the internet, different guidelines, you will find different definitions. Basic is the same, only terminology or presentation is the different. So method validation means what are the method you are developing So method development will be easy. Just you have to make the separation of the compound and you can finalize that. But you have to prove that method is suitable for your use. It is giving results which are highly accurate, highly precise. So that everything we have to prove during the method validation. So you can see the background or flow. Normally in the R&D lab, method development activities completed, and then it will goes for validation. In industry, pharma industry, there are two types of validations are there. Some companies are having method validation in R&D itself, and they will transfer the method to respective sites. Some organizations, they are following method validation at plant itself, so that method transfer is not required. So method transfer, it is short like technology transfer. So once you develop the method, you have to validate that method, and then that method has to be transferred to respective sites. It may be one site, it may be multiple sites. So we can transfer the method to different multiple sites, and then finally it will goes for approval. So any molecule, any new molecule you are developing, any new molecule you are planning to submit for Indian market or any other market, along with the data, you have to file validation, method validation data also, and all method has to be validated, like dissolution test, impurity test, potency test, or any other test like peptide characterization or identifications. So each method has to be validated, and then data has to be submitted to the regulatory authority. Then regulatory authority, they will review all the data and they will certify, yes, this method is suitable for use. So this is the flow for method validation. So now why validation is required? Now I have the method development, I have the partial validation in during R&D center. Then again, why validation is required? So this is first for identification of source for potential errors. So once you start the validation, you will come to know what are the potential errors in future will come to know. So we can take the actions, we can implement the corrective actions for that, so that future that errors can be avoidable. To demonstrate that method is suitable for its intended use. So if you don't have the method validation, that means that your method is not suitable for regular use. So that's the reason we have to validate the method. Then to establish that the test data obtained was consistent, reliable, and accurate. So what are the data we are generating by using this method? That should be the consistency. It has to be give repeated reproduced results. Then it has to be reliable data, and data should be accurate. So there are different parameters are there during method validation. We can prove whether data is consistent, whether data is reliable, or it is accurate. That I will explain in the next slides, coming slides. It is a regulatory requirement. So each and every regulatory, they expect method validation, whether it is India, whether it's Europe, whether it's US, Australia, Canada, any market. So it is mandatory requirement. Then to establish proof that method can be used for decision making. So what are the analysis you are performing by using this method? So that has to be decision making, means you are going to release finished product in the market, which is consumed by the patients. So that is the big risk if something happens, and if you are giving wrong results, and if you are releasing the product, tomorrow something happen to the patients, it will be big risk for the organization. So we are going to take the decision for release of the product in the market based on the method what we have validated. So that's the reason method validation is very, very important. So what are the different parameters for method validation? Any idea? Anybody is having any idea about method validation? What are the parameters, or anytime you come across? Anybody knows? In audience, anybody knows? Okay, we'll go to one by one. So first it is specificity. Specificity, there are two requirements. One is selectivity and force degradation. So I will explain this upcoming slide. Then precision. Precision also there are two types, method precision and intermediate precision. Third one is linearity. Then range. Accuracy, or sometimes it is called as recovery. Then robustness. So different types of robustness we have to monitor, like flow, temperature, or composition of mobile phase, pH of the buffer solutions. There are many parameters are there. then limit of detection, limit of quantitation. So these are the basic parameters which are mentioned in the guidelines.
Raj, unmute yourself.
Sorry.
घाटे सर सर्वांचे माईक बंद करा.
Hello. So these are the basic parameters which are required as per guidelines. But in addition to this, considering the experience and latest requirement for regulatory, so there are some additional parameters regulatory has expected. That is like stability of analytical solutions, then the mobile phase stability. So these are the general parameters required for all the method validation. Based on the test requirement, these parameters will vary. So you can see this chart. So there are parameters at left side and there are different categories. Category 1, Category 2, Category 3 and Category 4. These are the different tests. Category 1 means it is the assay which is called as potency. Category 2 means it is impurity profile quantitative test or limit test for impurities. Then Category 3 it is depends upon the titration technique, and Category 4 for identification. So these are the different test parameters. So you can see these parameters, what are the applicable for assay. So normally specificity, precision, linearity, range, accuracy, and detection and quantitation limit. These are basically applicable only for impurities. This is not applicable for assay or identification test. So this is the chart, it is mentioned in the guidelines. This is the minimum requirement, what are the regulatory authority they have provided. So based on the experience during designing the protocols, each organization they have to take a call, what are the parameters we have to do it. You can do it additional parameters, no problem, but minimum this requirement has to be complete. So you can see the specificity. This is the definition for specificity. Specificity is the ability to access clearly the analyte in the presence of components which may be expected to present. So if you take one tablet, in that tablet we are having one, take an example of Crocin tablet, we are having the paracetamol is active drug, and to make the tablets we have to add lot of things like binding agent, granulating agent, so many excipients also there, lactose, magnesium stearate, microclissan cellulose, talc. So these all are called as excipients, or it is called also as placebo matrix. So in the specificity we have to prove that when we are analyzing the product paracetamol by using HPLC, so paracetamol has to be clearly separated from other peaks. So typically these might be include impurities, degradation matrix, or lack of specificity of an individual procedure may be compensated by other supporting analytical procedures. So what are the placebo matrices? What are the impurities that should be well separated from main paracetamol peak? So that is, we have to prove in the specificity. So you can see there are two types. One is selectivity and one is the forced degradation study. So selectivity means we have to separate out main active ingredient from others, like from impurities and for excipients and from degradation products. So that means only specificity if you complete, that is not useful for us. If you are developing any new products, you are not aware that product how it will behave for next 24 months or next 36 months in the market. We don't have that much stability data because we are developing new products. So to check that behavior of the product, so what we have to do, if you see the forced degradation, name itself, it is mentioned forced degradation. So forcefully we have to degradation that product. So in the product, we can add acid, we can add alkali, we can have the peroxides. So different compounds we have to add in the product to generate the impurities. So forcefully we have to degrade the product. So that we'll come to know how much degradation will be happens during stability study or during next 24 months or next 36 months or next 42 months, which is defined for the particular product. So there are different techniques for forced degradation: acid hydrolysis, base hydrolysis, oxidation. For oxidation, we can use hydrogen peroxide. We can use the light, UV light, or normal sunlight also. We can keep the product at high heat or at 105 degree or 60 degree or 80 degree. We can expose the product to higher humidity, maybe around 80% humidity. This degradation we have to target in such a way that the impurity should be generated in between 10 to 15%. So that we'll come to know how much impurities are there and whether that impurities are able to separate out by using our method. So if you prove in this method, my method is very specific to the particular compound. So my method is developed for paracetamol and it is specific to paracetamol. Plus we have to prove that my method is stability indicative. That means during the stability study also any impurity generates, our method is capable to separate that impurities. So no need to redevelop the methods. Otherwise, sometimes what happens if you are not proving this, after 24 months, you will get some impurities in the product and your method is not capable to identify that method. So that's the reason we have to do this forced degradation study. So normally, if you perform this specificity study, it will look like this. You can see this is the main compound peak. I took it one example, and you can see these other impurities are well separated from this main peak. So we have to prove this in the method development as well as method validation, saying that my method is very specific to this compound and my method is capable to separate out all the impurities which are available in this product. So you can see all the impurities are well separated from each other. So there are different types of some impurities. If you see this one peak, you are saying this is the one peak. But sometimes what happens in the one peak, there are another isomers also there or close eluting compounds also there, which will be mixed with this. But by using HPLC, you will not come to know whether there is single peak or whether there is a multiple peaks are there. So there are some other techniques are there. In the HPLC, there are two types of detector. One is UV detector and another is PDA detector, that is called as photodiode array detector. If you use that photodiode array detector, you can scan this particular peak and you can find out the peak purity. So if your peak is 99% pure, that means there is only one compound. There is no mixture of other compounds are present. So that is the one technique, or you can use HPLC with mass detector. So high performance liquid chromatography with mass detector. So that is called as LCMS. So in that also you can find out the mass of both the impurities. So whether it is single or whether it is multiple peaks are there. So mostly people are following PDA detector, photodiode array detector, so that you will come to know peak purity 99% or it is 70% or it is 80%. Normally, if it is above 95%, it is acceptable. So this is the one example I took it. So you can see this main peak is clearly separated from all the compounds. Are you clear about specificity and this forced degradation, or any question anybody has?
I would like to ask, sir,
Yeah.
two questions. Hello, am I audible?
Yes, yes.
What is the actual concept and importance of the forced degradation?
Forced degradation, see, if you are developing new compound, today I am developing new products, say for example, any tablet. So I am developing ibuprofen tablet, for example. So if I develop the tablet today and I can analyze the tablet today, it will pass. It will comply all the requirement. But I don't know after 24 months, what are the impurities will generate in this product? Because I don't have the time, I don't have the samples which is having 24 months old. Because during the shelf life of the product, there is a scope to generate the impurities or degradation of the product. Like normally, most of the people, they are using amoxicillin triadate antibiotic. So if you see the amoxicillin triadate, it will degrade very fast and it will convert into the impurities. So at initial stage, within 1 month, I will not able to find out whether impurities are there or not. But after 24 months, yes, there are the impurities.
Okay.
So forcefully, we have to degrade that compound. We have to degrade that product by adding acid, by adding alkali, or by adding hydrogen peroxide, so that impurities will get generated. And we have to prove that impurities are separating like this. So that is the basic requirement.
Okay. And sir, my second question is, what are the selection criteria if you are using specifically diode array detector in HPLC?
So diode array detector, you can scan the compounds. Normally, if you see, you can see this peak.
Which one?
Whether this is visible for you, impurity G 23 minutes I mentioned here. So in HPLC, normal HPLC, you can find only single peak will be visible. But if you scan that molecule on the PDA detector, that PDA detector will prove that it contains only one impurity.
Okay.
It does not have any mixture of any other impurity. Normally, in case of isomers, it will mix and it will come single peak. But if you scan on PDA detector, it will give the peak purity. My peak is 99% pure. My peak is 98% pure. That means that is only one compound. There is no mixture of other compounds.
Okay. Okay. Thank you, sir.
So another parameter, it is precision. It is called as reproducibility also. So the general definition is the precision of analytical procedure express the closeness of agreement between a series of measurements obtained from multiple sampling of the same homogeneous sample under the prescribed condition. That means you have one sample. From that single samples, you have to prepare multiple samples. Normally, it is 6 samples is required. So from one sample, you have to prepare 6 different solutions, and then you have to analyze that solution by using HPLC, GC, whatever technique you are having in the method. And you have to prove that all 6 samples are giving same results. It may be the 90%, it may be the 80%. So all 6 results should be closer. That is called as precision, or in short, in simple language, it is called as reproducibility. So there are 2 types of precisions are there. One is repeatability and one is intermediate precision. So I will explain that also. Precision should be investigated using homogeneous, authentic samples. So your sample should be homogeneous and sample should be authentic. If it is not possible to get the homogeneous sample, it may be investigated using artificially prepared samples or sample solution. You can prepare artificial, you can use the individual tablets, you can prepare tablet powders also. There are multiple ways are there, but sample should be single sample. From that, you have to do the different solution preparation. So precision of analytical procedure is usually expressed as the variance, standard deviation or coefficient of variation of a series measurements. So this means you have to prepare the 6 samples from one sample. You have to analyze these 6 samples. You will get 6 different results. So like, for example, one sample you got it 99%, one sample you got it 98%, one sample you got it 101%, one sample you get it 100%. Like that 6 results will come. So you have to calculate relative standard deviation for these 6 results. So there are different acceptance criteria. In case of assay, normally it is not more than 2%. In case of the impurity, it may vary, depends upon the percentage of impurity. If your impurity limit is not more than 0.1%, normally it is 10% percentage RSD we have to give. So that acceptance criteria depends upon the technique, depends upon the instrument, depends upon the impurity level. So you can see that there are 3 categories: repeatability, intermediate precision, and reproducibility. So normally precision means what? One analyst, he has to perform the 6 samples one day. He has to analyze the samples. He has to check the results. If results are satisfactory, then yes, that method is precise. But again, we have to prove that intermediate precision. Intermediate precision means same sample has to be analyzed by a different person by using different equipments, by using different day or maybe different laboratories also. So that we have to prove our method is suitable for use of analysis by different analysts also, different labs also, or different instrument also. If you see the market, take it example of HPLC, there are different make and models of the HPLC are available, like Shimizu or Waters or Agilent. Different makes are there, different models are there. There are many analysts also there. There are many columns also there. So for intermediate precision, what we have to do? We have to select different analysts. We have to perform the analysis different day, maybe next day or next to next day. We have to use different make of the instrument. So that method is capable to reproduce on the different instrument and by different analysts. And then we have to compare both the results of first analyst and the results of second analyst. So if you see, there are total 12 results will be there. First analyst 6 results and second analyst 6 results. So that is called as intermediate precision. So that is the presentation. Reproducibility express the precision between 2 laboratories. So My method is capable to work any laboratory, any analyst can use this method, or any equipment can capable to analyze this method. So this is also very important parameter, because if it is not reproducible, this method is not suitable for use. So normally, if you see, a minimum of 9 determinations are required, or in the general practice in the industry right now, this is the practice is following minimum 6 determination at 100% test concentration. That means if my assay is 100%, my product contains 100%, so I have to analyze that sample 6 times. The results will be close to 100%, like 99%, 98%, 102%. Then we have to calculate relative standard deviation, that is called as percentage RSD. That should be not more than 2%. Again, same sample has to be analyzed by different analysts on the different day, and that 6 results has to be similar again. Then at the end, we have to compare this first 6 results and another 6 results. So that 12 results percentage RSD should be within 2%. If it is okay, then that method is acceptable for use, and it is called as the precise method. Can I go further?
Yes,
Yes,
sir.
sir.
Okay. So now we have started the validation. Now it is proved that my method is very specific. My method is stability indicating, and my method also now precision also it is there. So my method is precise also. Now one more parameter there is linearity and range. So normally if you see the method, any method, they will mention sample preparation and standard preparation, whether it is assay, whether it is dissolution, or whether it is impurity. So they have to prove what is the sample preparation. So in the sample preparation, for example, concentration is 100 ppm. So that is my 100% concentration. So my method is having 100 ppm concentration. Then we have to prove my method is linear in all the range. So in this case, what we have to prepare? We have to prepare multiple solutions with multiple concentration, like 100% concentration is the 100 ppm. Then we can have the 50 ppm, we can have the 80 ppm, 90 ppm, or 150 ppm. So minimum 5 different concentrations we have to select, and we have to prove my method is linear throughout the range. So you can see the linearity of an analytical procedure is ability to obtain test results which are directly proportional to the concentration. So if it is 100%, 100 ppm, so in that case, 50% also we have to prove for 50 ppm. So that method should be linear. So that is the range. So we have to prove entire range of the method. So linearity you can use directly of the drug substance. No need to go for sample. You can use standard also for this. For example, if it is the ibuprofen tablet, no need to use tablet directly. You can use only ibuprofen working standard or reference standard. You can prepare different solution of that reference standard, and you have to inject in the respective instrument, and you will get the different results. So for example, I am having 100 ppm concentration, and it gives the response 10,000. So if I am preparing the solution of 80 ppm, then it has to give the response of 8,000 ppm. Similarly, if I am having solution of 50 ppm, it has to give the response of 5,000 ppm. It has to be proved. So to prove the linearity, once you have the data, so what you will get it? You will get it 5 concentration, and you will get it 5 responses. You can use the 6 concentration, you can use 7 concentration. Minimum 5 concentrations are mandatory. So based on that, you have to do the statistical evaluation. You can calculate correlation coefficient. You can calculate y-intercept. You can calculate the slope of the regression line, and you can calculate the residual sum of the squares. So these 4 parameters you have to calculate. So based on these 4 parameters, like correlation coefficient should be 1 or at least 0.999. If it is there, then yes, it is your method is linear. I think you will come to know by this. You can see the graph. So normally this equation, if you see R square, that is correlation coefficient, it is 1, it is showing. This equation is y is equal to mx plus c. I think the people who is having statistics, they maybe knows. So based on this calculation, you can calculate R square. If R square is close to 1, that method is linear. So you can see these 5 different concentrations, 1, 2, 3, 4, 5, 6. And here you can see the responses. So if you plot this graph, you can plot this graph by using Excel, or you can have the different softwares also. There are multiple expensive softwares. You can get directly these calculations, or you can calculate manually by using graph papers also. So this is the simple calculation: y is equal to mx plus c, and r square it is 1. So that means my method is linear. If my method is not linear, you will see this graph little cross. The points should not be on the line. It will go outside the line. So then this R square value will change. If it is below 0.999, it is not acceptable. So this is called as linearity. So my method is linear for all the range. So in going future, if I am changing any concentration, instead of 100 ppm, if I am changing to 90 ppm or 110 ppm, still it is acceptable because I had already proved in the linearity and range. So in that range, I can change my concentrations. So in future, whenever I require to change the concentration, yes, I can modify this. Is it clear, linearity and range?
Sir, I have one doubt, sir.
Yes.
If within the optimum concentration range, if deviation occurs, then how you evaluate R square? Are you changing again concentration?
No, it is not changing. If it is you are going for very low concentration, for example, your main concentration is 100 ppm, and if you are going for 50 to 200 concentration, then that means your method is not suitable for 50 to 200. Then you can narrow the concentration range. So you can go to 80 to 120. So your answer will be here again. You can see this is the minimum requirement, 80% to 120%. So 100 ppm is your actual concentration. You can have the 180, 190, 100, 110 and 120. Like this 5 concentration you can prepare and you can prove. But sometimes what happens if you are having 200 ppm concentration, it will goes out of range. So that means your method is not suitable for that much concentration. You have to reduce the concentrations.
Okay, thank you, sir. Okay.
So this is the minimum requirement. This is for assay. You can see for content uniformity, it is covering from 70 to 130%. This is different test. You can see the for dissolution testing, we have to cover lots of range because dissolution, there are multiple tablets are there. Different types of tablet, immediate release tablet, or extended release tablet, or sustained release tablet. So based on the release of the product, we can cover the range. So sometimes within 5 minutes, some tablets are giving 10% release. So you have to cover the range from 10% to 120%. So that is the minimum requirement. So based on the dissolution release, you can select the range. So here important is you should know your product first. What is the release I am having? So for determination of impurity, from reporting level to the impurity up to 120%. But normally industry practice, we are covering up to 200% level. So this will helpful. Suppose in future, if my impurity is going above limit, still I can prove that. So still my method will be capable to analyze this product. So normally what happens at initial stage, impurity will be, you will get it impurity 0.15%. But after 12 months, after 18 months, that impurity will increase up to 0.2% or up to 0.25% because that is the degradation impurity. So in that case, I have to prove my method is capable to detect that range also. So we have to widen the range in that case. So that's the reason I told you should know your product better during the development stage only. What is the degradation I am having, or how much assay will be dropped during the stability study. So that much range we have to cover. This is the importance of the linearity and range. So now one more parameter it is there. It is called as accuracy, or sometimes we can call as recovery. So this accuracy or recovery, so I will show you the definition, then we can go further. So the accuracy of an analytical procedure expresses the closeness of agreement between the value which is accepted either as conventional true value or an accepted reference value and the value found. I think it will be difficult to understand, but I will explain you in better way. So accuracy, what we are having one tablet, say example again for ibuprofen tablet, it contains ibuprofen maybe 100 milligram, but it contains other excipients, many excipients are there. So what we have to do it, we have to prove, so in the presence of that excipients, I am able to recover my actual compound. So that means 100 ppm or 100% if I am having ibuprofen in my one tablet, whether my method is capable to take out from that 100. Or sometimes what happens if you are doing the analysis and if your method is not accurate, you will get the result 60% or 70%. So in that case, there are two possibilities. Either your product is not good or your method is not good. So in that case, we have to first check it during validation. Yes, my method is capable to recover from the placebo matrix. If I am getting 100% results, yes, my method is capable. If I am not getting 100% results, then I have to change the method again. So normally accuracy again, like linearity, we have to prepare here 9 determinations. The general practice is we have the 50% concentration, 100% concentration, and 150% concentration. Each concentration we have to prepare in triplicate. So all put together, you will get it 9 results. Again, these 9 results, we have to check it whether it is close to 100% or it is varying. So in the preparation, I am having placebo matrix. In that placebo matrix, I will add 50% of my drug, and then I have to prepare the samples, and that samples to be analyzed on different instrument. So if I am adding 50%, then definitely I have to get back 50%. If I am adding 100%, then it has to come back 100%. So similarly, all the levels it has to detect. If it is 50% coming back 50, that means the method is capable to detect all the recovery. So it is normally reported in percentage. Now here you will get it better idea. So you can see this graphs. So this is my target actually. This target means it is the 100% value. If it is goes above that, that means it is not close to the target. So you can see here different arrows are there. So that means the results are scattered. So it shows that my method is not accurate and my method is not precise also, because different results are there. You can see one value here, one value here, one value here. You can see one value this side. So there are different values are there. So only one value is close to the results, which is not acceptable. So my method is not accurate or my method is not precise also. This is the one example. You can see another example. Here you are saying all the results are close, but it is not near to the target value. So it is outside the target. But it shows my method is precise but not accurate. That means I am not getting accurate results, but it is reproduced results. Still it is not acceptable, because only precision is not acceptable. Method has to be precise as well as accurate. So one more example you can see. Here method is accurate. Most of the results are close to target, but it is not precise. You can see the results are scattered again. So this shows your method is accurate, but it is not precise. So in this all 3 cases, our method is not acceptable. So method has to be accurate, method has to be precise. First example shows method is inaccurate and imprecise. Second example shows method is inaccurate, but it is precise. Third example, it is accurate, but it is not precise. If you see here, it shows your method is accurate as well as precise. All the results are close to target and all the results are close to each other. So that means all the results are 99 or 98 or 101%, 100%. So this is the acceptable. That means my method is accurate, my method is precise. So if this is the results we are getting, we can accept this method and we can go further. Is it clear now, accuracy and precision?
Sir, I have one question.
Yes, sir.
What is the logic behind figure 9 in the accuracy evaluation? Is it based on standard value, standard deviation value?
There are some statistical calculations are there. For statistical calculation, for example, my percentage RSC limit is not more than 2%, then we have to collect the data from 5 populations.
Okay.
So based on your percentage RST limit, there are statistical calculations how much results we have to collect. So minimum 9 results are required based on the statistical evaluations.
So 9 is not the constant, it is changeable.
You can increase that.
Okay.
You can go 12, you can go 15, you can go 20 also, but minimum 9 is required.
Okay. Okay. Thank you.
Similarly, linearity also, in case of linearity, minimum 5 levels are required. You can go 6, you can go
6.
7, you can go up to 10 also. Not a problem.
Okay. Okay. Thank you.
The minimum requirement as per statistical evaluation. So now this robustness, or sometimes it is called as ruggedness. So robustness of analytical procedure is a measure of its capacity to remain unaffected by small but deliberate variation in method parameters, and provides an indication of its reliability during normal usage. So if you see any HPLC method, high performance liquid chromatography method, there are different chromatography conditions are there, like flow rate 1 ml per minute, column Mohn temperature 25 degree. You are having mobile phase concentration buffer and acetonitrile, or buffer and methanol 500 ml, or you can have the buffer pH maybe 4.5 or 5 pH. So what we have to do in the robustness, we have to deliberately variation these conditions. You can apply the flow rate, you can change the flow rate 0.8 or 1.2. You can change the column Mohn temperature like 20 degree or 30 degree instead of 25. You can change the buffer of the pH of the buffer solution. If it is actual pH is 4.5, you can go for 4.2 or 4.3. You can go 4.6, 4.7. You can modify the composition of the mobile phase. If it is 50-50 ratio, you can go 60-40, or you can go the 45-55 also. So depends upon the modification. So by modifying this, we have to prove that still my method is capable to give the accurate results. So for example, if I am having the flow rate 1 ml per minute, so your pump will be the give exactly 1 ml per minute, and you will get the retention time, you will get the assay of 99%. If I am keeping the flow rate 0.8 ml, still my method is giving 99% results. That means if in future any changes happens, my method is giving perfect results. So these are the conditions we can modify. There are many parameters are there for different methods. Like for dissolution apparatus, you can see normally we are using dissolution media 900 ml. You can put the dissolution media 890 ml or 850 ml. Purposefully you have to modify. So you can change the temperature of the dissolution media. If it is 37.5, you can go for 37 degree. So if you modify this and if you are getting the good results, yes, then your method is good, that is. So these are the typical variations you can have. So for example, extraction time, if your method, it is mentioned sample has to be mixed for 15 minutes or 20 minutes, or sample has to be sonicate for 20 minutes. So you can change that time and you can prove that my method is still capable by changing the time. Because what happens normally, if there are multiple analysts are there in the laboratory, there are many laboratories, 100+ analysts are there. Each time different analyst will come. So if one analyst, he's prepared the sample, maybe his shaking procedure will be different. Another analyst, his shaking procedure may be different. By hand, I am shaking the volumetric flux, it will change. By changing that also, if methods are giving good results, that means your method is robust. Any person can use the method, and we can prove that also. In case of liquid chromatography, typical examples, you can change the pH of the mobile phase. You can change the mobile phase composition. You can use different lots of columns or different supplier of the columns. You can vary the temperature. You can vary the flow rate. So like that, there are many conditions are there. In titration also, you can change many parameters. In UV spectrophotometer, you can change the wavelengths also. So there are many parameters. So generally, this is only to prove the method is robust. Now this one more parameter, it is limit of detection and quantitation. So limit of detection and limit of quantitation, it is not applicable for assay test or not applicable for dissolution test. This is generally applicable only for impurity studies. That is called as related substances or related compounds or impurity profiling. So this, you can see the name only, limit of detection. That means my method is capable how much detect I can do it, whether it is 0.1 ppm or 0.2 ppm or 0.5 ppm. So normal definition, you can see the detection limit of an individual analytical procedure is the lowest amount of analyte in the sample which can be detected but not necessarily quantitated as an exact value. So that means you can see the small peaks, how much I can detect by using this method, whether it is 0.05 ppm, whether it is 0.1 ppm, or whether it is 0.2 ppm. So this means only detection. It may not be quantitation. It may not be correct. Second parameter, it is quantitation. So quantitation of an individual analytical procedure is the lowest amount of analyte in a sample which can be quantitatively determined with the suitable precision and accuracy. The quantitation limit is the parameter of quantitative assays for low levels of compounds in sample matrix and is used particularly for determination of impurities and or degradation products. Now in short, detection means what? How much lowest concentration my method is capable to detect, and how much lowest concentration my method is capable to detect accurately. That is called as quantitation limit. In case of detection limits, you will see the peaks by naked eyes, but you will not get accurate results. You will not get reproduced results. So sometimes what happens, you will get the concentration, for example, 0.1 ppm for detection limit, but you repeatedly analyze, you will not get 0.1. Sometimes it will get 0.05 ppm, sometimes 0.15 ppm also. So that means your method is not capable to quantitate. So for quantification, again, you have to find out the concentration. Normally, if my detection limit is 0.1 ppm, your quantitation limit is 0.3 ppm. So that means my method is capable to detect and quantify up to 0.3 ppm level. So below that, my method is not able to detect accurately. So that is the maximum I can go at the low level. So I'm having examples, so you will get it in good idea in that. So there are many methods to determine LOD and LOQ. Normally it is called as LOD, limit of detection, and LOQ, limit of quantitations. So there are different approaches also there. You can see the based on the visual evaluation. You can see the smallest peak by naked eye also. Based on the signal to noise ratio, normally for limit of detection it is 3:1, and for limit of quantitation it is 10:1. In previous days, like 10 years back or 15 years back, this we should be calculated manually, but nowadays all advanced techniques are there, so softwares are capable to give direct results. So one more technique, it is based on the standard deviation of response and slope. So I will explain that in next slide. Based on the calibration curve also you can calculate this. So these are the different techniques. You can do it by visual evaluation, you can do it by signal to noise ratio, you can perform by standard deviation and slope, and you can take it by calibration curve also. You can see this diagram. Normally this is the chromatogram. This is the main peak of your interest, maybe impurity or API. This is the noise you can see. This is the baseline noise, it is called as how much noise I am getting. So I am clearly seeing peak here. You can see the how to calculate the noise. We have to take it lowest and we have to take it highest, and this is my main peak. So there are different calculations are there. You can calculate 3.3 into sigma divided by slope. Or you can have the limit of quantitation by calculation 10 into sigma divided by slope. This is the slope method calculation we normally called as. Or you can calculate by manually also. So you can see this heights you can calculate, and you can calculate the noise. So you can calculate signal to noise ratio 2h divided by h. That is 2 into h means height of this main peak. You can see this main peak, and h means you can see the noise of this. So you can calculate this noise level also. So 2 into height of main peak divided by h, height of the noise. So that is called as signal to noise ratio. This is the manual calculations, but nowadays you can get this all calculation through softwares. So no need for manual calculations. So up to this quantitation, these are the basic parameters which are required by all the guidelines on all the markets, whether it is European, US or Indian market. In addition to that, based on the experience of last few years, now regulatory agencies, they are expecting some additional parameters like stability of analytical solutions. So what we have to do it, we have to prepare the solution, standard solution as well as sample solution. We have to keep this solution at room temperature, and we have to keep at a different temperature like 5-degree temperature or 10-degree temperature, depends upon the compound. You can store these solutions for 1 day, 2 days, 3 days, or whatever you require, 7 days, 10 days, depends. Every day we have to analyze this solution again, and we have to calculate the results. So based on these results, we have to see that how much days solution is stable. Some solutions are stable up to 72 hours, some solutions are stable up to 24 hours, some solutions are 5 hours, some solutions up to 10 days, depends upon the molecules and depends upon the diluent what you have selected. So how much solutions you are getting stable, so you can mention that solution stability in your respective test method. So during quality control analysis, that analyst, he will come to know that my solution is stable up to 24 hours or up to 36 hours. So I can use that solution up to 36 hours. If today I am preparing the solution and it is stable up to 36 hours, yes, I can use same solution tomorrow also. I can use same solution day after tomorrow. Till 36 hours, I can use that solution. But that has to be clearly mentioned in your test procedure. What temperature and how much time. So normally what we have to do it, we have to analyze day 1, that sample and standard. Again, after day 2, again we have to analyze, we have to compare the results with day 1. After 2 days, again we have to analyze, again we have to compare with the initial analysis. So like that, if you want to prove up to 7 days, you can prove up to 7 days. You can go up to 10 days also, nothing stops you, and you can mention that. So normally it is calculated in percentage difference, initial versus next time point results. It may be up to 10 days or it may be up to 7 days or 2 days. And we have to prove our solutions are stable up to how much hours or how much days. So similar type, we have to prove mobile phase stability also. So I am preparing the mobile phase. Today I prepared the mobile phase, for example, 5 liter mobile phase I prepared. Normal flow rate is 1 ml per minute. So in 24 hours, how much mobile phase will consume? It may be around 1 liter. So I can use that mobile phase for 5 days. Again, we have to prove that data by analyzing the different days and proving that system is stable up to 5 days. So you can use that mobile phase, but we have to check physically also, because sometimes what happens, mobile phase contains buffers and organic solvents, so chances of precipitation is more. So if it is precipitate, then we have to throw out the mobile phase. So this is called as solution stability. Now we have completed the method validation, and my method we have given to the quality control team for routine use. So now again there is one more criteria when revalidation is required, or whether I can use that method lifelong, next 50 years or next 100 years, or I have to do the revalidation. So there is some criteria for revalidation. So you can see, if you are changing the synthesis of drug substance, then yes, you have to go for method validation. If you are changing the composition of finished product, that means you are changing the formulation, you have to go for method revalidation. If you are changing the analytical procedure, you have to go for revalidation. The criteria for revalidation, it may not be go for complete revalidation. So you have to do the risk assessment. What are the parameters? I have to go for revalidation. So you can see how much composition I am changing, or what the synthesis, route of synthesis for API we have changed. So based on that, we can decide how much revalidations. So these are the references you can see. ICH guideline Q2R1, validation of analytical procedure. United States Pharmacopoeia, Journal Chapter number 1225, validation of compendial procedure. Guidance for industry, this is for US market, Q2B, validation of analytical procedure. European guideline, sorry. These are the guidelines we are having. So if you see, ICH guideline will looks like this. I don't know, I'm able to open. Can you see this?
Yes, sir. We can see this.
Yes, this is called as ICH guideline, International Conference on Harmonization. So if you goes for US guidelines, it will look like this: guidance for industry, Q2B validation for analytical methodology. So if you want to European guideline, you can see this EMEA guidelines. So it will look like, but most of the guidelines now parameters are same. So nothing change will happen. So that is not a scenario for 20 years back, because there are different guidelines, different criteria was there, but now it is entire harmonization they have done. So that's all from my side. If any questions, I'm open now.
Sir, I have 3 questions.
Yes, sir.
In your one of the slide, you have shown that robustness can be evaluated and how it is evaluated. But often robustness is different than the ruggedness, which is also the another criteria parameter for method validation.
Yes, ruggedness, what I mentioned is repeatability, intermediate precision and repeatability, that is called as ruggedness.
Okay. My second question is, if the bias value is encountered, then how you overcome and evaluate in method validation?
If bias value is observed, so you have to just first do the investigation, whether it is error from any analyst, whether it is error from any instrument. If it is not, then you have to check the method. You have to redevelop the method again.
Okay. And my last question, when and how the method is partially validated?
See, if I am having the method validation at one site, for example, in my lab, method has been validated and I am having method validation data. And if I want to transfer this method to other sites, so then I have to provide them complete validation protocol and complete validation reports. So based on that, they can do the partial validation, 1 or 2 parameters in that lab, and they can continue to use that method. No need to go for complete validation again.
Okay. Thank you, sir. Thank you.
Thank you, sir, for your detailed lecture. Sir has explained all the basic concepts of analytical method validation. Thank you very much, sir, once again. Now, if anyone have any kind of doubt or question, please ask.
You can ask in English, Hindi, Marathi. I am comfortable with 3.
Sir, can you share this presentation?
I can share.
Okay, sir. Thank you.
Mahesh Ranchur has any problem? Mahesh, have any question? Any doubt? Mahesh Ranchur?
Actually, virtually it is quite tough to explain. If it is offline, we can explain better, I think. But next time we'll see.
Yes, sir. We will organize offline one lecture. Now, I would like to invite Professor Mendele, sir, to express the vote of thanks.
Hello, am I audible?
Yes, sir.
Yes, sir.
Okay. Honorable Resource Person, Mr. Animesh Salunke, sir. Respected Advocate Gautam Patil, sir, who is the Chairman of Janta Shikshan Mandal Alibag. Respected Principal Dr. Anil Patil, sir. Vice Principal Dr. Sonali Patil, Madam. Respected Head of the Chemistry Department and Convener of today's national webinar, and our colleague, Professor Ashok Jadhav, sir. My all departmental staff members, on behalf of organizing committee as well as JSM College Alibag, I feel very immense pleasure to take this opportunity to propose a vote of thanks to our today's eminent resource person, Mr. Animesh Salunke, sir, who is being with us as a Vice President and Site Head of Invitron Advanced Analytics Private Limited, Hyderabad, who spare his most valuable quality time to grace this today's national webinar with a really and extremely relevant address. He very much rightly pointed out so many facts within just 1 hour about the method validation, which includes need of validation, parameters to be considered while method is validated, as well as last not but the least, the importance of revalidation also. So I thanks really Mr. Animesh Salunke, sir, for being with us with a very good lecture. I especially express also my deep regards and gratitude to our Honorable Advocate Gautam Patil, sir, for encouraging us and providing an opportunity to arrange today's national webinar, which is a part of academic activity. I would like to especially thanks to our beloved principal, Dr. Anil Patil sir, as well as Vice Principal, Dr. Sonali Patil madam, for all types of valuable guidance and support for organization of this national webinar. I would further extend a heartily thanks to our head of the department, Ashok Jadhav sir, for his entire enthusiasm, entire efforts for organizing this academic activity. My sincere gratitude also goes to our entire organizing team and all the departmental staff members for taking the topic that is really a matters today, especially pharma industry is concerned and the relevant era of analytical chemistry is concerned. My deep sense of appreciation and thanks to all non-teaching staffs of our department for their help and cooperation for conducting this webinar. Last, but surely not the least, I thank all the student participants also who chose to be live online with us and attended this national program with a great enthusiasm and made it as a successful academic event. Thank you all of you for being with us and have a wonderful day. Finally, on behalf of entire organizing team, I declare that today's national webinar is over. Thank you. Good day.
Thank you, sir.
Thank
Thanks
you,
a
sir.
lot.
you, sir.
Please
Thank you,
take a screenshot. So start your the